introduce, I started at MIT as assistant professor about 2 years ago. So my lab, the core expertise is that we apply bioengineering approaches and biophysical approaches to understand the human health and then develop corresponding medical applications in a mechanistic and quantitative manner. The key piece of information that we bring to this community is how drugs are transported using the principles of drug transport phenomena and fluid mechanics. And some of the examples of what we study, we are a very broad group. So we study the different disease mechanisms. So that's the pathophysiology part. And we also study how we can model different systems better so that we can develop better diagnostic tools. And last but not least, coming to what I'm going to talk about today is how we can apply the knowledge towards designing different therapeutics. And then as also Manuk introduced, my lab has both a wet lab as well as a dry lab component. On the experimental side, we focus primarily on in vitro assays and some ex vivo assays. Most of the assays are microfluidics, and then you will see some of it in a sec. And then we combine it with our own computational modeling tools. So we really think that the two can go hand in hand together. And then my students, whether they're experimental or they're computational, they talk to each other a lot as well. But ultimately, for any health-related applications, we do need to make sure that it indeed works in a complex biological system, that even combining the two together may not be enough. So we are going to work with different in vivo collaborations. And being in the Boston area, although MIT, my home institution, doesn't really have a medical school, but there are a lot of different medical schools, especially in the Boston area, that we work with. And I'm also eager, if anyone have any exciting problems, feel free to come talk to me, and I'd be happy to establish other collaborations with researchers as well, elsewhere as well. So the key knowledge coming from my presentation today is that, first of all, we focus on microcirculation. So we focus on small blood vessels that are sub-millimeter sized. And then here, we already saw presentations talking about how there are different circulating blood cells, and each of the circulating blood cells corresponding to different diseases. Red blood cell, if you have abnormal red blood cells, that can correspond to sickle cell disease or malaria. And then we have white blood cell that can correspond to inflammation, which can be a good thing or a bad thing. And then you have platelet, as we saw from the speakers yesterday, it's really a balance, whether it's going good or bad, it can lead to hemostasis or thrombosis outcomes. But then inside this blood, there are other components as well. And when things go bad, you can also have circulating blood tumor. The one that I'm going to talk about today is actually how to use blood for how to understand blood from the perspective of drug delivery. But by no means we're treating all these different 5 components in blood as independent and non-interacting. In fact, you will see over and over in my talk today that we're actually going to design drug delivery systems based on a lot of the information that we learn from these 3 types of cells that are already present in the blood. Okay, so here is a general agenda for what I'm going to talk about today. Given this is a lecture, I'm going to start with an introduction about how blood circulation is related to the drug pharmacokinetics. And then I'm going to move on to moving from free drug molecules to a nanomedicine form and how that is going to change the pharmacokinetics. And then I'm going to talk about how nanomedicine is used for thrombosis directly treatment, and how the in vitro and in silico models, as the title suggests, and also what our group is doing, and as well as showing you some examples from other research groups from both aspects. And then we can do actually more things than thrombosis treatment. Nanomedicine can actually be used for thrombosis-inspired applications, taking the knowledge that we learned there. And eventually, I'm going to move on to the forefront, which is the current ongoing research in my lab, as well as others, on how to combine nanomedicine and cell therapy, where a lot of the cells here correspond to the circulating blood cells to achieve more complex missions. Okay, so let's get started. So the first topic I want to introduce, if you do not come from a pharmacy background, is that during drug development, if we identify like a molecule that is going to be effective and also having the least side effects to other parts of the body, It does not stop here. So the drug development process is much more complicated than just considering the efficacy and side effects. Those are called the PD, the pharmacodynamics. And for example, if you know what molecule that you're going to use, which route you are going to choose for the drug administration. It can be oral. This is a pill that maybe you're taking regularly. This is the most convenient route. But the challenge is that anything that you take orally, they need to first cross the GI tract in the gut system in order to enter our body. And then during this process, a lot of them are lost. So not all of them will actually enter our body, despite the fact that everyone just like to take pills. And then you have nasal route, and this is a route where you have highly permeable vasculature. So first connection to the vasculature, the blood concept here. But the problem, as you can see, how many of you actually take any drugs that are nasal? The development right now is quite limited, and there are a lot of limitations there. Another one is the ocular route, because the eye is one of the very few organs that are directly exposed to the outside. Most of the other organs are located inside our body. And here in this schematic, this is referring specifically to the eye drops here that you can apply. This for eye diseases are great, but if you want the drug to go to other side part of the body, as you probably have never seen such things before. But most of the things you see in the hospital, as well as your medication experience, is injection, and mainly injection into blood directly. That is the most traditional route, and that is the most common route as well. Although everyone hates needles, but this is a route that is commonly used over and over. Maybe we'll hear something at the tour in the afternoon as well. And then one of the considerations when people are choosing this route, we're using the same molecule, so there's no difference in terms of the efficacy and the part, but it is actually about the pharmacokinetics, the PK. So PK is the time course that is describing how a drug is going to move into our body, throughout our body to different locations, and eventually maybe getting out of our body. So a big consideration from this slide is that when you're considering how to design a drug, you need to consider the movement of the drug, and that is the PK. And then let's dive into the PK a little bit further. So as I said, intravascular injection, specifically intravenous injection, remain the most popular route. And one of the key plot that you will see in any PK studies is how the concentration of drug in your blood plasma is going to change over time. In the case of intravenous injection, immediately you will see a spike because that's where the drug is going to, and that is going to decay with time. Versus if you choose the other routes, you will see that it won't be immediately available there, but it's going to increase and then decrease. So here are some of the considerations when you look at these for your PK and drug design. First is that we always see this decay over time. But if we see a decay, meaning that the drug concentration is reduced in blood, doesn't mean that it is going to the site where you want it to act. So say if you wanted to treat like a lung disease, is it actually going to the lung? The answer is unfortunately most of the time no. Your best good approximation is probably if you take a volume average or mass average of all your organs and just assume that everything is going to be evenly distributed, you can calculate how much is actually going to the site of interest. But actually some of the times you can get something even lower than that because there are other mechanisms that the body is going to get rid of the drug. Next one is that if you look at the area under these curves, are they going to be the same? Meaning that if you inject or if you use different type of drug administration routes, are they going to eventually deliver the same amount of things to blood? The answer is also a no, because these drugs can also be metabolized, so they can degrade over time, etc. So in many of the cases, if you inject 100 grams, or even using other drug administration routes to administer 100 grams, what you actually get is way less than that, even considering blood, before it goes to any side of targets. So there's huge losses there. And the last one is, do we want these changes to be fast? Or do we actually want it to be a very slow decay, etc.? Well, if you have something that is really acute, maybe you want it to be really, really fast. But for most of the cases, people actually do not like to see these kind of fast decays. Because if you have a very fast decay, that means in order to achieve a certain dosage that is going to be really effective, not to be like a small enough dosage that's going to be just negligible, you actually need to keep injecting, having multiple of these peaks in order to make sure that the area under is always going to be something meaningful. And that is the concept of controlled release. Okay, so let's take a look at how the drug exactly moves. So the passive physical mechanism for the drug to move inside our body is through a combination of evacuation and diffusion. So if you recall from your physics classes, diffusion means that the drug is going to move from higher concentration to lower concentration with the goal of eventually homogenizing the whole system. And evacuation can actually occur from lower concentration to higher concentration because it only follows the direction of your fluid flow. And one common fluid flow that we'll encounter in our body is the circulation, where if it's inside the blood, it is going to follow the evacuation scheme there. But while this is doing these transport, and also, by the way, this is the passive physical mechanisms. These days, you can actually have active ones where you're actually like using magnetic force or other things to actually give them an additional kick. But once the drug transport of the drug from one place to the other is actually not an easy thing. So it actually need to cross multiple of these what people call the biological barriers, or if you come from like a chemical engineering community, that is essentially the resistance to transport. So this is athletes competing in the Paris Olympics Games for the hurdling competition. And the drug is essentially doing the same thing because it's not going to reach its final destination from the site of injection all smoothly. It needs to cross a lot of the barriers here, as shown. So just give you an example. If you inject it, the first barrier that you will have is that you need to make sure inside the blood, it is not going to be uptaken by any enzymes or by the white blood cell here. So this is the first barrier it needs to be overcoming. Second barrier is that it needs to somehow at some point cross the endothelial cells, cross the blood flow, and then enter the local tissue. So that endothelial cell is another barrier. And once you cross that, you also navigate through the extracellular matrix that is present in your local organ system, and that can vary from one organ environment to the other. Last but not least, even if you're super close to the cell that you want to target, if the drug mechanism acts by having the drug acting in some units that's inside the cell, you also need to be able to cross the barrier and enter the inside of the cell. So this is just an example of all the barriers that you could possibly think of as you have the drug, how the drug is transported to one site to another site of interest. And given that we're talking about blood here, at least for the first two steps, they're highly related to everything else that is happening in the blood system. And by the way, when the drug is trying hard to cross all of these barriers to reach the site of action, remember that there's also the competing process, that's the renal clearance. So essentially, the kidney system is going to like to get rid of all of these drug molecules. So as you're crossing the barriers trying to get to the site of interest, this is actually a much easier to cross because this is a body system that is just trying to naturally build to get rid of these. So you also need to compete with something that seemingly to be very easy to happen. So that's the reason why for a lot of the free drugs or the drug molecules, purely say in a solution form, etc., it is very, very difficult for you to get a desired concentration that is in the site of interest, and then not having excess concentration anywhere else to have any side effects. But even if they go to other sites without any side effects, you're still losing a lot of the drug molecules. So that's how there's a new concept emerging that is nanomedicine. It is a very simple idea that we want to now give the drugs an additional carrier to help them to better go to their site of action and then increasing their favorable distribution in the site of action and reducing their distribution or concentration elsewhere. Here is just a review paper highlighting some of the very common ways of designing these nano carriers. And the nano carriers are very broadly designed in the sense that it can be in a particle form, or it can be more in these polymeric forms that are essentially the size in the same nanoscale size. But some of the things you have in common is that you have these yellow parts, so those are the actual drugs. And then you're designing them with a different structure, a lot of them containing these blue and red part. And those are usually functionalized units, helping to achieve a purpose of increasing the target efficiency. And some of the things to consider here, first of all, these narrow carriers need to be designed with biocompatible materials in order to achieve effective clinical translation. Some of the common ones you see are PEG and PLGA, and actually there's not a lot of materials that are biocompatible. And we already saw a great talk from yesterday's presentation. And the next one is that, so you do need to know these properties very well to be injected. And you also need to make sure that before injection, they can be stored stably for a very long shelf time. And during that process, it can be in the powder form. So it doesn't have to be in a solution form before you do the administration. And last but not least, if you replace all of these yellow particles that I said previously represent drugs to some contrast agent, the same concept of using these nanocarriers can also be done to actually do in vivo imaging, where you're essentially using these carriers to help you better image any site, any organs of interest. And probably posing a question here is that why do people do nanomedicine? Why don't do something that is bigger, etc.? Well, it turns out that the question, if you ask people, the answer will keep getting updated. And I think the most latest answer that I can give from my perspective is that the nanomedicine's definition has broadened a lot. These days, people know that in certain cases, you actually want to go to like a micron size in order to have it perform better. But one of the things that you will see over and over in my presentation today is that the size does matter. Are they similar to the size of a cell, or are they something smaller? There are always these different considerations and the reasons. And there are still some aspects that we do not understand very much. Okay, so now let's go to the next section about how the examples of how to use nanomedicine. First of all, for the case where if you only have one barrier to outcome, that is just let it stay in the drug, stay in the blood, and then treat any of the diseases related to the blood itself. So let's look at these what people call the vascular targeted nanomedicine, specifically for this conference for the thrombosis applications. So there are many of them, and I'll only show you a few. This is a polymeric nanoparticle application actually developed by my colleague, Professor Paula Hammond. They're a polymer group. So what they designed here are these polymeric nanoparticles, and they design it in two different forms. One is just a purely like a standard polymeric nanoparticle form, where this polymeric nanoparticle is going to have this targeting component that is going to target the platelet. And actually, this is also an interesting case because you actually only have the targeting part of your nanocarrier design. You actually don't have like a real drug that you're delivering. But on the right-hand side, here you also see these additional component. So what it does is that this new carrier design actually have another part that is used for cross-linking, with the hypothesis that the targeting component is going to be beneficial for treating primary hemostasis related to the platelets. And then you have the second component that is going to be helpful for treating secondary hemostasis, with the hope that treating the two together is going to help you to facilitate forming a more stable clot to treat internal bleeding. And then another highlight that I really like from this paper is that you always need to be careful that when you inject these things to the bloodstream, it is not causing any other side effects. For example, is it actually triggering clot formation in the site that you do not want? So they carefully designed it so that there's going to be a threshold concentration that only above which that this is going to have this secondary mechanism kicking in for forming the stable clot. So below a certain concentration, it is not going to be activated that way. So that is going to help us from preventing unwanted clot formation at other sites. Okay, so this is one method that was shown to be effective in vivo. And let's look at another example. So this is a liposomal type of nanoparticle that is used for also improving the thrombus formation. And this is for treating von Willebrand factor disease from Professor Anirban Sengupta's group. So here, what they actually have on the surface is 3 different type of peptides instead of 1 that is for targeting purpose. So you have 1 type of peptide that is targeting the activated platelet. You have another peptide that is going to target the binding to the endothelial cell layer once it is exposed during. And then the last one is a VWF binding one. So instead of having 1 type of targeting, you actually have these 3 different ones shown as different colors. And again, for the treatment or the encouraging the clot formation, you actually don't need to encapsulate inside these nanoparticles any other specific drugs. You just need to have these surfaces that are functionalized in these ways to promote the clot formation. By learning from this paper, what you can actually deduce is that if we have more fundamental understanding of how clot is formed during different conditions, how different diseases are going to change the pathway of binding, you can actually use those to develop these multiplex carriers where it is going to not only have 1 type of targeting, but also have different ones to be able to serve different purposes, essentially filling in the blank of what is currently missing in the clot formation process to achieve a 1 + 1 greater than 2. And this is different from if you just have 3 different type of nanoparticles where 1 particle is having only the blue peptide, another one is yellow, another one is green, and you simply just mix them together. Because you know that when you have the 3 of them combined together, based on our understanding of clot formation, this is going to help us to promote all of them tying together because we know that this is a process involving all of these aspects. Yes.
Just a short question. You mentioned this barrier to which have to be overcome. How is this how you can assure that the particles don't be attacked by the immune system?
Yeah. So
why they are invisible for our defense units?
Yeah. So one of the most common use of the PEG is to actually achieve that problem, and biocompatible materials will also help to mask that, etc. It depends on the problem. But I'm also going to show you that one of the unknown things is actually how they're coupled together. But this is the first time that I'll show something related to an in vitro study. So you guys already saw very many wonderful talks regarding the use of coated flow chamber to study any type of clot formation mechanisms. So in this paper, you're studying the action of these peptides on these platelet adhesion mechanisms as well. So the use of in vitro systems here compared to the previous paper where they focused just on the animal mice studies is that you can actually use these in vitro studies that are carefully designed to help you understand the mechanism better. And that's going to help you really in the field of if you want to keep advancing, you're not just satisfied with, okay, one set of experiment in vivo, it seems to work, then can we actually use these in vitro systems, very different parameters, and also design the control experiments carefully to see are the hypotheses of how they work really work the way that you want. And then the next generation of in vitro models is going to go beyond just having a coating of different kind of molecules, collagen, von Willebrand factor, etc. It is actually going to have the real endothelial lining, as we saw from the talk yesterday as well. So this is another example of a microfluidic device. Here you have the whole blood, and then you have the vessel walls that are lined with endothelial cells. And then you can promote injury by having this tiny little slits where you're essentially going to punch on it, and that's going to trigger the bleeding. And when you use these endothelial cellized systems compared to the previous systems where it's just a coated surface, you need to consider more biological complexities here. This can be a good thing in a sense that you're really going to, like, if you want to move to patient-related studies, as we saw from the discussion yesterday, you want to really make sure that anything that is captured, defects from the endothelial cells can be captured by this, and that is possible to approach these days. But then on the other hand, once you move to these endothelialized systems, your experimental cost and your time and the error bars are going to change significantly. In terms of the experimental time, the previously said the coated flow chamber experiment, those are very fast experiment. And we already saw from the mention yesterday that usually within an hour, you can do even multiple sets of experiment. But for these, the reason because the blood, you can just directly collect the blood and flow them through it. Versus in this case, once you need endothelialized systems, you need to actually culture the cells inside the flow chamber for a period of time, wait for them to mature and being functional again, and then start to do your experiment. And that time scale is you first need to culture them in the flask for however many long time, then you transfer the cells to your device, and then you need to culture them for at least 3 days. And then your cost is going to increase because here the material of the microfluidic system is also going to change. In the sense that previously you can use very synthetic systems, but here you do need to have PDMS because you need to have the cells here to be breathable. So you need to put these in an incubator, 37 degrees C, 5% CO2, etc. So all the material requirements are going to be higher. So are the error bars, because now you're using the real cells. It's not going to be as wild of a distribution compared to the in vivo study still, but you do need to repeat your samples for like increase the sample size in order to make any argument about the statistical significance. So now I'm going to show you another exciting example of using these endothelialized systems to study drug delivery applications. So this is from one of the groups at Michigan, where what they do is that the surface is endothelialized, and also they're damaging the surface by both using chemicals as well as a physical damage using a scalpel. So that's why you see these places that are more concentrated. And then this is a system where they designed a new kind of polymeric particle to help reduce the adhesion of platelet leukocyte aggregates to the site of injury that is related to thromboinflammation that you will hear a talk tomorrow as well. So there are some very interesting findings from it. First one is that when the particles are targeted, meaning that they're functionalized with specific units on the surface, versus non-targeted, meaning that it's just a purely passive particle that is biocompatible, both cases are observed to help reduce the adhesion of these aggregates in vitro. So that means you need to have at least 2 mechanisms action for it to work. First one that's easy to think about is that the targeted ones, they do have, in this case, instead of targeting the platelet binding, here they focus on targeting the binding between the particle to the endothelialized surface. So they're now targeting the endothelial cell layer. And that is the active part that they're putting in. But there's another important part that is why the passive particles also work. And that they put in a hypothesis that it is because if you look at very close to the endothelial cell layer, there are white blood cells, there are platelets, and there are also these passive nanoparticles that are injected. So the hypothesis is that the use of the particles is actually going to increase the chance of hydrodynamic collisions between the particles versus the platelets and the white blood cells. And due to these additional collisional events that's happening near the endothelial cell wall, you will have less likelihood for the platelets and the white blood cells to aggregate together as compared to the previous case without the particles. So you're essentially adding something that is disturbing the formation through hydrodynamic collisions. But this, they did not do a direct testing of it. Why? Because the setup of this experiment is looking from the top down at the endothelial surface. And in order to be able to make any argument about this part, you do need a system that is actually going to look probably from a different angle to validate that. And that is something that's very difficult to achieve through these experiments. And that's why I'm going to introduce to you the computational models that you can do for this process. So computational models are essentially, instead of in the experiments, you make these phenomenological observations about things, and then you come up with hypotheses about these mechanisms. Hopefully that you can design some control experiment to validate by knocking off different things to see whether this is indeed the case. Here, we can build these computational models where we take a bottom-up approach, where we're going to capture the biophysical and biochemical cues that we think are important, incorporate them through the mathematics, and see whether we are able to generate the desirable outcome and then compare with the experiments. And on the other hand, if once the models are validated, we are excited about using them to make new predictions to help facilitate the experimental designs as well. And when you talk about modeling, the modeling can be done at different kind of what we call length scales, but I will give you a very high-level description of it. So at the smallest length scale, or when you zoom in to whatever that's happening inside the blood very carefully at the molecular level, you can do these molecular dynamic simulations. So each of the particles here, if you want to, can be modeled with these explicitly modeled green particles. Those are actually going, each of these spheres are going to represent one unit of a ligand. And then a lipid bilayer, as shown as these red and the yellow spheres, are going to be modeled using this hydrophobic and hydrophilic different units as well. And then you can move up in your length scale, and then you can go to the mesoscopic level modelings. So this is the models where instead of directly resolving all of these specific things, you're just going to model these different cells, and each one of the cell can be modeled either as a rigid entity, meaning that it is just something that you can calculate its XYZ position versus time, or you can model them by doing a discretization into hundreds of mesh points so that you can calculate them in terms of shape changes so that overall together you can get a shape contour, etc. And then finally, you can move to the continuum level, where in the continuum level of blood simulations, instead of directly resolving the dynamics of each single cell, you're essentially taking it as a continuum distribution, and that is how these two are related. And we also saw some great explanations by Professor Fogausen yesterday. So as you move from left to right, as you move up, you are looking at different levels of the details that you're interested in. Of course, this is going to give you the most complete information, but in order to resolve anything that is big of a size, this is going to be very, very costly, because here you're talking about things that is instrument level all the way to here that you can talk about things that's millimeter length scale. So you're easily talking about increasing the computational cost by orders of magnitude. So one of the takeaway here is that you need to pick a modeling method that is going to make the most sense in terms of resolving whatever the detailed physics that you want to resolve, but at the same time, making sure that this is still not going to be a very expensive simulation that is going to take years to accomplish. And coming back to the previous question from the experimental hypotheses, that they hypothesized that the particles are going to have collisions with the white blood cells and the white blood cells and platelets near the endothelial surface, you do need to have a model that is probably able to resolve those collisions. So if you want to do it that way, probably the mesoscopic level is the right approach to do. Okay, so in my PhD work, I work a lot on this mesoscopic level simulations, and I'll just give you a quick overview of it. There are many different ways of doing it. You can do boundary integral method, you can do lattice Boltzmann, you can do DBT and inverse boundary, etc. But what they share in common is this, and they are able to resolve. Now, when you look at this picture, instead of the experimental picture where you're looking top down at the surface, here you're looking at this from the side. What it means from looking by the side, and actually you can change the rendering from your computational system so you can look at it from all these different three-dimensional angles. But this video is showing you from the side. So all of these red are the red blood cells. This simulation shows the white, which are the platelets, but the modeling of the drug particles, etc., are done exactly in the same fashion. So what people can calculate from these simulations are the shape changes if you want to model any soft cells, the positions of them over time, and also if you're interested in the stress field of the surrounding fluid, etc., there's a lot you can calculate as well. What people can actually validate is the formation of the free silico layer due to the cell migration. I want to just re-clarify these concepts. So red blood cell migration means that red blood cells are soft entities. Even due to complete physical aspects, without considering even anything biological, due to the fact that they're soft, and they are going to have a tendency to be pushed away from the cell wall. So they don't like to stay close to the blood vessel wall. And that is going to leave the near wall region as a cell-free layer. That's the free silico effect. And then margination, that's another concept that applies to platelets as well as the drug particles we're considering. Because red blood cells occupy such a high fraction, or the hematocrit here in the system, all the other components are usually existing at a much lower concentration, so they're the minor species in the mixture. And because of that concentration difference, they are going to be pushed by the red blood cells essentially towards the side, and having a near wall concentration excess that is called a margination. And margination is also related to the platelet adhesion as well, because when you measure the platelet concentration from a bulk sample during the blood test, that is assuming that this is a very well mixed mixture. But even at that overall concentration, when you look at what is actually the specific concentration near the wall, it is going to be significantly higher due to the margination effect. So that also means you cannot just use a bulk platelet concentration to make an easy argument about whether there's more cells or not. So I could have one bulk concentration that is higher than the other, but due to the change of the red blood cell concentration, I can have actually lower platelet concentration near the cell wall compared to the other case. And then why do we care about this? Especially, usually the cell-free layers are on the order of micron. When you talk about microcirculation, where the blood vessels are small, these cell-free layers are going to be comparable to the size of the blood vessels you care about. Versus if you study bigger blood vessels, then the cell-free layer is so small compared to the size of those blood vessels, so that their effects are usually considered not to be that important. Okay, and then there's people think that computational simulations will usually just run very fast, click a button, and then you can get a result very quickly. But then this is actually one example where computer simulations may not be as fast as you think. The problem is that, as I said before, red blood cells are very soft. You can put in these different constitutive equations to model the shape deformation of the red blood cell. But the problem is, instead of calculating just the movement of one single point, as in the case of a rigid particle, you actually need to calculate hundreds of mesh points in order to resolve the shape change. You cannot describe any shapes with a single point. So due to that, the computational cost is significantly increased compared to just simulating multiple rigid particles. So that's why these simulations can be a pain in the neck to simulate. Aaron made an argument yesterday, and I can relate because I did a lot of simulations in my PhD that was actually running for like 4 to 6 weeks. So you do need to carefully design the simulations in order to make sure that you're not going to be generating just garbage data by the end of that time. But we also work on one way that can reduce the time of the computation. We're not actually reduce the computational cost overall by that much, but the idea is that instead of simulating hundreds and hundreds of red blood cells, we are going to break it down into simulations consisting of only one cell or 2 cells. And these cells can be either cells or platelets or particles here as well. So by breaking down the big simulation into smaller pieces, we can run multiple of these in parallel. And that is going to be a good way of reducing the simulation time from 4 to 6 weeks to, in some cases, 1 to 2 weeks. And then we also validated that if what you care about for the red blood cell contribution is simply just what is the red blood cell distribution in the cross-flow direction, so that you can use it to help you calculate what is the platelet concentration near the wall or the drug particle concentration. We validated that using this reduced order model approach, we can actually get a good fit with the experimental data. So red and blue shows 2 different hematocrits that we did the prediction. Okay, so one of the other things, as I said, is that you can simply easily add nanoparticles to these simulations. One of the things I also said is that you have these different sizes. So how does the size of the nanoparticle play a role? So one of the things you also need to consider is that the smaller the particle that you get, you also need to consider the role of Brownian motion, and that is going to be more and more of an effect for smaller particles. And Brownian motion is going to help the particles to actually be more homogeneously distributed. So here is what we predicted for the particle distribution across the channel. So the wall is at the left and the right. You will see this margination behavior with the near wall axis. But as you go to order 10 nanometer rigid spheres, as shown by these black dashed lines, you will see that the margination is already less pronounced compared to the 100 nanometer as well as the 1 microns. So that means if you purely consider from the perspective that I want to have more particles near the walls, then probably you don't want to go that small. And then coming back, there is also this term that is in the equation. This is a term exactly corresponding to the original experimental hypothesis about particle or particle cell collisions. So that's the part that's also resolved by these simulations. Okay, and then in these simulations, you can also add different adhesion dynamics considering the binding kinetics, and the binding kinetic modeling can go on and on depending on how many steps you want to model. And we already saw a great presentation yesterday by Aaron, so I'm going to skip that. But I want to just show you one example where these addition of the adhesion dynamic simulations can bridge the in vitro and in silico models even further. So this is one, actually it's not about drug delivery, it is just about platelet adhesion in whole blood. So there's also red blood cells in the system. So every bright dot here is one platelet. And when you run these experiments under different flow conditions, as well as changing the hematocrit, you're going to get different behaviors for the patterns of adhesion. And then normally you would just calculate the rate of adhesion from this, and then we can use the simulations to do the same thing. But the problem is, if you want to do a diagnostic tool, where in diagnostics, you need to make sure that because there are multiple different causes that can change the rate of adhesion. Is it coming from red blood cells, or is it actually showing in this case where we're essentially reducing the receptor density of the GP1B factors on the platelet surface by using AK2? And instead of in many other experiments where you do a high concentration of AK2 to completely knock out all the GP1B, we actually carefully choose very low concentrations so that we can see a gradient of a little bit knockout, 50%, 75%, etc., where at each one of them, we're going to see different patterns. So if you want to tell purely from the K-on value, the rate of adhesion, whether this patient has a hematocrit problem or whether this patient has a GP1B problem that can be related to Bernard-Soulier syndrome, you wouldn't be able to make an argument. But if you do further analysis of these images, based on the understanding and based on the modeling that you can do to model these multi-step processes, you can have not only just one rate constant, but multiple rate constants. When you have multiple rate constants, it can help you to better distinguish these different kind of adhesion behaviors. And same concept applies if you want to extend it to drug delivery, platelet adhesion, vascular targeting as well. And using this, we also came up with different other rate constants that I'm not going to show you, but they can help you to differentiate further behaviors. And one of the things that our collaborators at Royal College of Surgeons, they did initially when they had more than 200 people's videos like that, is that can we just watch the videos and then hire a company to do a data mining and then come up with some phenomenological parameters to do it. It turns out that because there's, even within the healthy patients, there is such a wide distribution of the videos that you can see that we actually published a paper talking about it, that those phenomenological definitions and modeling to predict these behaviors is actually very sensitive to the way that you arbitrarily set any parameters. Like, how do we even define whether the particle, the platelet is stationary or not? Depending on how we define it, the result can be sensitive, and that means it's not a really robust modeling. It is only when we join force together, and then we realize that instead of thinking about things from the top down, we need to think about things from the bottom up. then we hope to have more robust system to account for these differences. Okay, so now I'm going to switch gear a little bit and now begin to talk about how we can move away from directly using nanomedicine to treat clot formation to something that is inspired by this process. Okay, and here we're talking about at least whether we can have something that is going to involve the crossing of the endothelial cell barrier. Okay, one of the examples that I'm going to show you first, in vivo and in vitro combined study is this microfluidic device where they are growing these blood vessels that are inside a microfluidic device. And then alongside that, you also have the glioblastoma tumor. So you have a tumor spheroid combined with the blood vasculature in the same system. And then using this system, they validated the drug delivery mechanism for designing some glioblastoma targeted nanoparticles that you can see from these red signals, and using the in vitro system device, almost like how you would do an in vivo imaging to actually track how the nanoparticles are going to move around in the system. And one of the interesting things here is that this is a disease regarding the endothelial cell layer, so the in the brain. So that's forming the blood-brain barrier that is known to be very tight. But in many conditions, people actually use the nanoparticles with the assumption that during disease conditions, you're going to have angiogenesis and the blood vasculature, the blood vessel wall, the endothelial cell lining there are going to be more permeable. And that is a condition that's going to be favorable for nanoparticles to cross. Versus in this case, they actually proved that even when you have a very tight blood-brain barrier, they actually did other studies to show that there's no leaky vasculature here, but properly designing the glioblastoma targeting, they're actually still able to cross the barrier, like a white blood cell in some sense. And but although what they showed in this study seems very promising, that is both in vitro as well as in vivo, there are many issues that are surrounding the use of nanomedicine. People doubt, is it really going to work or not? And one of the review papers that was kind of controversial in 2015, published by Warren Chen's group. Okay. Yeah. Is I want to highlight this part that they essentially did a review of nanomedicine publications produced in the last 10 years from, so it's 2005 till 2015, is that if you really do a careful analysis for how much of these administered nanoparticles actually go to a solid tumor, the answer is around 0.7%. What does that 0.7% mean? If you take like an average mass of a solid tumor and consider dividing that by the overall mass of your body, you will probably get a number that is even slightly higher than that. So this is even worse than if the drug is homogeneously distributed, and you're claiming that you're using nanomedicine to make it better. And the number actually didn't even change much after 10 years. So he was raising a lot of concerns on whether this is going to work, at least for the case of solid tumors. And then he proposed this map of how we should study nanomedicine from the fundamental above again, and to really make sure that we carefully do the design to make sure it actually works. Some of the things that we can relate to the blood community, he did highlight that we need to study nanoparticle blood interactions with a deeper understanding. He also highlighted that we need to perform these foundational studies to actually guide the design, which, as I said, we can design a lot of in vitro and in silico experiments to help us as well. Another thing is that he mentioned once we select our optimal nanoparticle design, so I already showed you a few different surface chemistry, et cetera, that can be tuned. So there's a lot of tuning knobs you have. Once you optimize, you can go to the large models, and then you may need to probably go back through the process again, because once you know something works for mice, and then when you translate to a different animal model, is it going to work the same way? Because, well, even for red blood cells, I can speak that the mice red blood cell and human red blood cells have significant differences in terms of the size and the rigidity, different, reduced volumes, different, etc. So you cannot guarantee that the optimal design for one animal model is going to work the same for the next animal model. So there's going to be a lot of iterations, etc. He proposed actually at 2025, we're going to be at this stage, but honestly, I don't think we're anywhere near there yet because a lot of people in the drug delivery community actually just ignore this map. Let's just keep going and then see whether it works in vivo or not. And then if it works, let's come back to some fundamentals. If it doesn't, ignore it, etc. And but at the same time, even when there are concerns about nanomedicine, there are people moving forward saying that, well, at least can we come up with a different kind of concepts where we are going to actually use some of the knowledge that we learned from circulating cells, especially coming from this community, to help us at least do a more informed design where even we don't know how the nanoparticle would interact with the blood. We know how the blood cells interact with the blood. So can we use it to do something better? So a new emerging concept is to actually combine these nanomedicine, mainly nanoparticles, say, with natural cells so that you're going to form these hybrids. And the most popular candidates are these circulating blood cells. To date, white blood cell, red blood cell platelets have all been used for this demonstration. The eventual goal is that you probably take a blood draw and then you manufacture these using the patient's own cells and then send them back to the body. That's the vision. And then one of the things that, excuse me, that people find that is even exciting is that depending on the cell of interest, and you can tune the physical chemical properties of nanoparticles in different ways so that you can achieve different outcomes. So for example, say we think about particles that are attached to red blood cells. Why people design it that way? Because red blood cells have a very long circulation time versus if you look at the drug concentration versus time curve that I showed you very early, you see a quick decay. And I said, this might be just everything is quickly get washed out for by the kidney. So if you use the red blood cells, the nanoparticles carrying the drug can be hypothesized at least to be staying in the blood circulation for a longer time instead of being quickly get rid of away by our body's own defense system. But what's even more interesting is if you change the number of nanoparticles to red blood cells, you're actually going to get different outcomes. So depending on the ratio that you're chosen, how many of these nanoparticles go to the lung versus how many of them go into the spleen can actually be tuned. Anyone can guess the reason here. So remember, red blood cells are very deformable. These particles, you can design them to be soft, but they're never going to be as soft as your red blood cells. So when you combine them together, is it going to make the red blood cell more rigid or is it going to make it more soft? More rigid, right? And where do the more rigid red blood cells get stacked? Spleen. So depending on how you tune it, you're essentially mimicking an aged or rigidified red blood cell or something that is closer to the mechanical properties of the original red blood cell. So there are actually papers published where people are traditionally just simply choosing the tuning knob of how many particles are you adding to the system to actually help you direct the drug to have more distribution in the lung for lung type of diseases or targeting the spleen for spleen type of applications. So this is compared to if you just have the nanoparticles, if you want to achieve the two different outcomes, you need to design the targeting to different cases, or even if you want to target the lung, maybe it will still go to the spleen, etc. So here you're using these fundamental principles that we learn from blood to actually design it in a much more informed way. But there will be some in vitro challenges and in silico challenges as well. So I'm about almost an hour in, and usually when I teach, I know that students have a very limited attention span. So I will always stop here for a break, and I'd be happy to take any questions here before I move to the ongoing work, as well as some of the challenges that I see in the current work. Any questions? Yes.
Thank you very much. It's impressive. Just a curiosity. The nanoparticles are injected are supposed to be injected into the patients, right?
Supposed to be
Injected.
yes.
Into the patient. But at the moment,
injection is the most common route, although there are work being done for other administration routes, like say eye drops. There are eye drop nanoparticle formulations that are like startup companies, etc. So it doesn't have to be through this route. I'm focusing on the intravenous ones because for intravenous ones, you actually, for the other routes, people talk about the barriers. They will spend more attention to maybe other barriers they're going to cross, but I'm focusing just on the intravascular one so that I can focus on the blood aspect for this audience.
Thanks. Super. And so far, there is already like some clinical trial, or is still at the animal testing level they use?
And there are some that are even failed companies.
Thank you so much for the talk. I think it's very interesting. I think your approaches are very interesting. Also, for my questions about the actually thrombosis inducing drug delivery, for example, von Willebrand factor disease. So I was thinking when you want to induce thrombosis, can you also do it like locally to treat like aneurysm or something? Or would you say for something like that, it would be more beneficial to have like drug delivery probably by coating of the flow divider or something like that? Could you specifically target aneurysms for thrombosis formation during drug delivery by nanoparticles?
Yeah, well, I'm not an expert on that, but I'm an engineer. And my short answer to it is that if you could provide me a molecule that you know is going to act in that fashion, then there are ways that people can work around to package them into a nanoparticle form. So that's why, coming back to my earlier point, is that we need a lot of these fundamental understandings about which sites and which molecules we need to target and what we need to use to make it work. Then engineers can help to make it happen.
Maggie, this is awesome. This is a really good overview of the state of the art in drug delivery. I was wondering if you could help pull together two parts of the talk for me. You started talking about pharmacodynamics and pharmacokinetics. And I'd be sort of curious, these are lots of different particles, and I know it's hard to generalize, but I imagine the pharmacokinetics are significantly different than small molecule or proteins. And in the context of, maybe on two ends of the spectrum, von Willebrand disease, which is maybe treated prophylactically, what would that look like in terms of, half-lives even? I think that's always a nice, I think an easy to understand concept, just what's the half-life of these particles? And then, how would you envision in the context of thrombosis, which is generally a surprising and an unpredictable event, right? So unless we know somebody is at risk for thrombosis, they probably don't have nanoparticles circulating around. So maybe just your opinion on those things, because you've thought about this more than I have.
Yeah, so coming to the first part regarding the PK or like the half-life of the process. So that's something that can be tuned by like in the nanoparticles. A lot of the designs are actually not just on, okay, you have a shell and then you have the inside. You can do like layer by layer or having extra hydrogel layers as a resistance to essentially delay the transport of the drug from inside to the outside. So that's a way to actually be depending on how you design it. And then you can also change the cross-linking, etc. So even with the same layer thickness, you can get different rate at which it is getting out. So using these are the tuning knobs that you can tune the half-life. And then as to whether we have the range that is capable to capture whatever the application is needed for, that is a question that I think needs more of the careful design, like even in vitro model, in vitro and in silico models to help, especially this doesn't even need to have probably sometimes a purely like you don't need a blood vessel, maybe you just need a blood plasma, and then do careful like release studies, etc. So that's one. And then regarding your second question, that's a great point. And then for the diagnostic tool on microfluidics that we were working on previously, the idea is that people, whether you're like normal or you already have an underlying disease, you do these blood tests often. So if you can you use these assays as a tool to actually predict the risk of something happening and then administer some drug based on the risk. It's not like this person already have something that is happening, then we start to treat it. So actually using it as a predictive tool, and then that's why Army was interested in it, is that can we have a database of every soldier's adhesion profile so that we can quickly pull it up and figure out what is the best strategy, either to do it right during the comeback, like right before that happened, or something that can be immediately administered. So that's one of the concepts that I think I completely agree with you on the challenge that probably giving people nanoparticles all the time is not going to be feasible. But then combining with the use of in vitro and in silico tools, that's actually going to be in a more proactive fashion in terms of predicting what's going to happen in the future. And then to make best intervention at that point is probably something that is worth considering.
Can I ask you about delivery of the nanoparticles? Do you see them being injected and taken out by red cells? Or are you talking about what they're doing in Oxford? You're growing up your red cells in the lab and you fill them with nanoparticles and then you put the red cells in the individual. Which way forward would you choose?
So the design of this one, nanoparticles stay on the surface, and then there were characterizations that were being done by proving that it doesn't go inside. But I'm actually going to talk about the ongoing work very soon, where staying on the outside versus going in is actually another tuning knob. And then how to tune it is something that we're working on to predict what is the criteria for going in or out, so like staying on the surface, so that people can better use it too, depending on the scenario that is of interest. Does it answer your question?
No, I said in Oxford, red cell.
Yeah.
Could you pre-label the red cells in the laboratory and then inject the red cells, or do you prefer to prepare your nanoparticles and give them to an individual so they're taken out by the red cells?
Yeah, gotcha. That's a great point. I think that both approaches have been used. And I actually see there are in the US, there are companies doing something similar to what you just mentioned as well. So both are possible as long as there are like, as long as the culture is known to be like an effective process, even for generating just functional, normal, healthy red blood cells, to we make sure that there's no other effects, etc. There's actually, I believe that there's actually Boston company that is doing the same thing as well. So I think that both are possible. And I do want to clarify that to make sure that these things are not going to be causing significant disturbance to your body, you will never have them existing at a very high concentrations. So if you look at how many are the engineered red blood cells versus how many are the original red blood cell, calculated percentage is always going to be very small.
Thank you very much for your inspiring presentation. In the context of nanoparticles used to treat the internal bleeding or to help to treat the von Willebrand factor disease, do you think that a not perfectly designed number of nanoparticles can induce thrombosis? And if yes, how do you think that we can test the perfect number of nanoparticles that we need to use for treat the, for example, von Willebrand factor disease, but not induce thrombosis?
Another very good question. So that is something that people don't know the answer. That's something that's part of the 30-year plan that was proposed by that controversial paper, but nobody actually followed. I can tell you that I think in silico models are going to be tremendously helpful, that I'm going to show you one example very soon too, so that you can, at least if it's not the 99% prediction, you can at least use it to narrow down the amount of experiment that you're going to try, because it's going to definitely have very high confidence predicting what is going to not work. So you don't need to try all the different parameters, as you just mentioned, all the different sets of experiments. So it's going to help you reduce. And then carefully designed in vitro experiments are going to help you advance a little bit further as well. So that the hope is that you're going to use this combined approach to help you significantly reduce the amount, so that you're going to really design it in a very careful way to help you get the critical information. If it's not the optimal, at least close to optimal.
Last question. Thank you for this great studies. Actually, I just heard about the nanoparticles is attached onto the surface of red blood cells, right?
Mm-hmm.
It can also go, it can also go inside of the red blood cells, I guess.
Depending on the regime, and I'm actually going to show you one study about, for red blood cell, it's going to be challenging, but for platelets and white blood cell, it's actually easier.
Yeah. Actually, I have a further question about it. Will it affect the hemoglobin binding capacity if the nanoparticle is attached onto the surface of red blood cells?
Well, I'm not aware of a study, but that's something that we can probably look at together.
And okay,
one question.
Is it able to detach from the surface due to the mechanical forces or shear stresses, etc.? Did you check?
Yes, that's gonna come up next.
So I guess you can continue, and then we'll finalize the session of question after.
Great, cool. So now I'm going to go to some ongoing work that I frame it as challenges and perspectives altogether. First one is that as you now have these entities that are having both a cell component as well as nanoparticle component, the mechanism for you to test for why it works becomes more complex. So how do we design experiment that can understand these rather than just purely a nanoparticle, how it's going to act? For example, one of the hypotheses from in vivo studies is that nanoparticles, once they're loaded onto the red blood cell surfaces, will require, once they're in the small blood vessels, to actually have contact with the endothelial cells and then forming adhesion, etc., for if you want to do vascular targeting. So that's kind of already related to what you just meant. So we designed this microfluidic experiment where these are all endothelialized, and then you have these red blood cells that are attaching, carrying these polyphenol functionalized complexes that are some in the nano size, some are in the micro size, and they're fluorescent label. So all the green you see correspond to these nanoparticles. So what we see on the flow is that they do attach. And this is a microfluidic device that is much bigger than the small sizes that they're claiming. So even in bigger blood vessels, you actually can still have the nanoparticle to adhere. You don't need to really go to the smallest ones in order for it to happen. So we did further analysis of the black and white signals here. So we didn't label the red blood cells, but because we have a lot of red blood cells, this is a whole blood experiment. You can see from the gray shaded area, actually a rough estimation of the red blood cell distribution. So here, the top and bottom, and then again, now our device is changing its setup a little bit. So it's different from the previous adhesion assay that you're seeing a different angle. But looking at this angle, you will clearly see the endothelial layers are at the top and bottom. And then you see these green highlighted regions that you don't have gray shades. So that's under normal conditions, that's the cell-free layer, the first linkage layer effect. Versus if you do the same thing for these complexes that has red blood cells carrying the nanoparticles. And here we didn't really mimic the physiological condition as I described. In reality, you will have a mix of the healthy red blood cells with these ones. But in this experiment, we have just purely all engineered cells. When we flow them together, we actually see that the empty region becomes in the center. You have great shady areas that is all the way up to where the endothelial cell layer is. So what this actually tells us is that now the red blood cell, because from only looking at the green images, you only know where the nanoparticles are. But this wouldn't tell you actually whether the nanoparticles are still with the red blood cells together or they have already detached from the red blood cell. So by examining a second color, we're able to prove that essentially the nanoparticles are still attached to the red blood cells. They all together are moving to the side, forming these adhesions. So they're almost like a platelet. So that's why I say this is thrombus-inspired, although you're now engineering red blood cells. But if you don't know how it works for platelets, it will cause a lot of issues here. And then another challenge that we're facing is that if you want to look at any processes where it involves crossing the endothelial cell layer, or even consider the next step of the barrier, that is how it interacts with the local tissues, you need to have essentially the endothelial cell layer, and then you need to have something crossing, which in a lot of the cases that also involve immune regulations. For example, in this case, this is from our collaborator that they actually validated that the blood-retinal barrier, similar to the blood-brain barrier, normally there's going to be, it's an intact barrier, and then it's separating the human retina of the human eye with this blood circulation. So the eye is considered to be like separated from all the other parts of our body. That's the immune privilege. But they actually find that under glaucoma disease models, you will be able to observe the circulating T cells shown as these green dots that they have infiltrated across. And that means, can we get inspired by these to actually have nanoparticles attaching to T cells, which have already been shown to work for other organs, to now have them infiltrate under disease conditions and use it as a way to treat glaucoma? And then they even found some heat shock protein 727, which is a molecule that is detected that is thought to be related to the process, where HSP27, coming back to thrombus, is also a pro-thrombotic factor. So all of these are in vivo experiments, and then build on that, they hypothesize a loop that when you have a bleeding or when you have a glaucoma, etc., that's going to cause neuron damage. Neuron damage is going to activate the local retinal microglia. That's going to cause the breakdown of the endothelial cell layer. T cells are going to infiltrate across, and then that causes more of the local microglia activation, more damage, and then leading to blindness, etc. So that forms a loop, and then only when you understand this loop in a much more informed fashion that you really validate each step and really making sure you know which are the molecules that are involved, can you actually going back to the question of designing your nanoparticles that way to know what needs to be the surface functionalized unit, what needs to be the drug inside, etc. And that is a question that is very difficult to answer using the conventional animal models. And that's why we join force together to develop even more advanced microfluidic models that are able to test the hypothesis. So then now we're moving from first generation, just coated layers for doing those flow coating experiment, to endothelialized surface, and then moving to the new generation where in addition to those two components, we also have something that is going to represent the local tissue so that we can really validate the entire process. And previously, there were some simple studies where they just have endothelial cell layers, and then they can have T cells or whole blood on one side, and then on the other side, they can add just chemicals or cytokines, and then due to chemotaxing, that's going to attract the T cells crossing the barrier. But what we want here is that instead of artificially adding chemicals to trigger the process and the study, can we actually mimic the system better by actually having cells on one side to trigger the study? And then I'm going to skip some of the experimental details, but happy to revisit it. But one thing I want to show you that now we're talking about experiment that lasts for 7 days. And then we have multiple different type of cells in the system, multiple different cells that you need to culture. Previously for endothelial cells, you just put the cells into the system once. Now for these more complex systems, you might need to put in multiple different types of cells to the system. And then all of these cells do not grow at the same speed. So another underlying factor that people often omit when developing these is that you can't put everything into this device at the same time. So there was a lot of error, trial and error at the beginning of figuring out how to put them into environment. And another even more complicated problem is that they may all, if you read the protocols for how to grow these cells, they need different kind of food for their growth. But when you have them in these kind of devices, are you making sure that they're compatible with each other, similar to the hemocompatibility problem that was illustrated yesterday? And then we managed to finally overcome a lot of hurdles to actually show you this structure that was my first slide. So essentially on the right-hand side, this is a confocal showing you the cross-section of the blood vessel compartment where you have these endothelial cell linings. But most importantly, you really need to make sure you have this vertical one, because this is actually the transport barrier that is going to actually determine whether you have the cells crossing from the blood side to the left side, that is the local retina side, or not. So just the top and the bottom are not enough. You really need to make sure that you have this part. And then on the left-hand side, you only have a monolayer structure, that is the human microglia. And these days we have an even more advanced model that I don't have a confocal image, that we have neurons on this side as well. So I will skip some of the details, but I really want to show you this video here, where this video is showing you the movement of white blood cell. And you can imagine in the future, these are going to be the drug delivery carriers. Each one of them, or some of them, will carry the drugs as well. So they're fluorescent labeled. You can see their movement from the right to the left. And then there is a squeezing motion of these T cells that is undergoing the infiltration due to the fact that you have something on the left that is not a chemical, but a real cells that are releasing whatever the chemicals that is needed to making it happen. And this can become a tool that in the future we can use it to test whether any nanoparticle drug delivery approach are going to work or so, or even to study more complex problems. And I'll skip some of these, but I want to show you this one. So I showed you a video of the cellular movement, and this is something that we can already track more carefully compared to any in vivo studies, because they do not have that resolution to look at these. But we can actually generate new knowledge. One of the new knowledge that we generated from the process is that previously people think, okay, it's just about T cells crossing the barrier to the other side, then done. What we actually attracted is that there's actually going to be these interactions between the microglia and the infiltrated T cells shown in the blue color. And if you do a careful analysis of these, you want to look at that they are highly correlated in terms of this position. This is something that previously people do not have any knowledge. So using in vitro systems, you can not only just try to mimic an in vivo system and do these mechanistic studies, but also even generating new knowledge, because in vivo currently cannot be accessible to that image resolution. But there are other challenges, and I want to offer as perspectives, and maybe some of you will work on something like this in the future. First of all is that in these in vitro systems, it is very easy to have a leaky blood vessel. It might not be leaky from just bare eye, but it is leaky at a microscopic level that is going to be more permeable compared to like what is actually happening in the body. And then that is a reason for why a lot of these microfluidic devices, organs on the chip, they do claim they have a blood vessel, but they do not use the whole blood for extended amount of time, as you already saw some of the issues from yesterday. So for a short period of time, this is totally fine. But if you want to observe it for an extended period of time, people normally do not use the blood, whole blood. And even coming to our study, we have neurons in our system. Once the blood is leaky to that vessel, blood is known to have toxic effect to the neurons. So that's going to directly kill the cells. So although you can think, maybe this is going to model some disease conditions, but the problem here is that we don't know what disease, what degree we're controlling. If we know it, then we can create like a disease model, great. But if we don't know how to control it, this is still a problem. And I highly encourage you, when you look at these papers, to dig into the detail. Are they using whole blood, or are they using something different? A lot of the studies, they actually just use one component. The video I showed you didn't have whole blood, didn't have red blood cells either. And another part is that you have typically, if you consider the concentration of these things in your blood mixture, it's very, very slow, very, very small. And the microfluidic devices are also very, very small. So that means you actually don't have a lot that you can track on each image for that small scale that you're considering. And that can have a lot of issues. And so in a lot of these studies, people actually intentionally use a much higher concentration of nanoparticles, such as this one here. Like you see a lot of nanoparticle signal. This is definitely not going to happen in vivo. People are going to die. So we are exaggerating the effect because we have a miniaturized system. but how to make sure that we understand how to scale it back to the in vivo concentration to make sure it is meaningful, to make any predictions, etc., that is still a challenge. And the last but not least is that the quantitative tools developed for these devices are still lacking, in a sense that the geometries become more and more complicated because we can fabricate different things. But that means a lot of the conventional analysis tools are no longer going to be valid. So even doing the image analysis here is no longer a problem that is no longer like as straightforward as previously when people analyze just a time series of images. And then on the other hand, we know that for pharmacokinetics, there are a lot of models that are being done for free drugs or solution form that can predict different things as in my past publication. So can we actually extend these into the nanoparticle or the particle aspect to do some models as this as well? For example, in this paper, the analysis of the nanoparticle signal is exactly the same way as you're analyzing the phytidextrins by assuming that everything is continuum. So is it really continuum or not? And then can we really take any continuum approximation to make sure that this is the meaningful validation or measurement? That is still a question. And then on the in silico or the computation side, there are also things that we need to overcome as barriers as well. The first one is that now we want to have a simulation where instead of the normal cells that we're studying, we're going to simulate the engineered cells. But the problem is these engineered cells consist of these nanoparticles that are way much smaller compared to the size of the cells themselves. Remember, I showed you the 3 levels of simulations you can do depending on the length scale that you're interested in. You're here interested in the cells, but you definitely have these much smaller objects that are governing the changes in the cell dynamics. So this becomes a multi-scale problem that a brute force way of simply just keep refining the numerical resolution is, given the problem that the simulations are no longer already very expensive, it's probably not a feasible solution. So how to capture these influences, or even think about how to rewrite the cell mechanics, the constitutive equations, accounting for nanoparticle as one of the function parameters, is something that is not known. And we are doing some project in this area as well. I just want to go to the findings, is that here, this is a previous prediction of a phase diagram corresponding to a case where you're assuming that the particles are infinitely dilute on the cell surface, which is a good approximation because you never want to have a lot of particles unless you have a application where, say, you want to rigidify the red blood cells and you want to target the spleen. But for most of the other cases, you still want something that is similar to original red blood cell. So this is each one of these colors correspond to one type of outcome that is going to predict, is the particle going to stay on the surface of the cell? Is it going to enter the inside of the cell, or is it not going to stick at all? So each one of the color correspond to different kind of outcome. Each one of the dot is essentially one experimental prediction. So you can see by doing these parameter sweeps under different parameters, you can essentially predict thousands of experiments by doing these predictions. But what we find is that as you increase the particle to only 0.1% surface fraction, instead of assuming that it's close to 0, so 0.1% is also negligible to many people, right? But the problem is you see the color has changed significantly. The color change for the phase map means that the outcome for the experiments that we predict are changed significantly. So the big take-home message is that we predict these nanoparticles, although they're much smaller compared to the size of the cells you're considering, and they're not existing at a high concentration, you can actually have significant changes in terms of the cell biomechanics using these particles. So that's another reason why people still need to keep working on this field, should not ignore their presence and their influences. More careful analysis needs to be done to make sure that we really understand the mechanism before moving forward. And as I said, there are other challenges as well. One is that, as you saw from yesterday, endothelial cells are shear dependent. So how can we properly model these in our system? And how can we model a leaky endothelial cell layer? How can we model the process of something squeezing through, whether it's red blood cell or white blood cell, etc.? Another one is parameter value. Once you start to do the experiments, you realize that not all the endothelial cells behave the same way in terms of adhesion properties, etc. That can be having huge implications. We are interested in it because for the eye research that we're doing, the human retina itself have two different type of blood vessels. Even looking at a bare eye, the cells look completely different. So how to know whether the parameter values we're inputting to the model are going to capture these biological specificities, that's another challenge. And I want to show you some common challenges that both in vitro and in silico needs to work on. First is, if you have a blood disorder, say in this case, a sickle cell disease, and if you want to design drug delivery carriers, now the original sickle cell disease is already going to change the distribution of the cells inside, and then you can see only a fraction of the cells are actually sickle cell, and then some of them are still normal, and then as you can see, their distributions are different. So that means what you previously predicted for how the red blood cells in the normal conditions are going to behave, to use that to predict the drug particle outcome, are no longer going to be applicable in this case. And when you move to these diseases, it's even more difficult to do the prediction because for every patient, it's going to be different. Healthy is 100% healthy red blood cell. Disease, you can have 10% of sickle cell, you can have 50% sickle cell. Every sickle cell, is it rigidified like this for only a small amount, or is a degree? That's a whole different question that currently there's not, like, given the computational cost, that is always going to remain a challenge, how to properly model this process. And also on the in vitro side, how are you able to make sure that you design these, how many in vitro experiments, how many different mixtures are you going to create in order to do these, and then how many patient samples are you going to use? Another challenge is to consider other components of blood other than the cells. We are now just considering a suspension where you have different type of cells and particles in plasma, and then plasma is just treated as just a Newtonian fluid and consider nothing else. But the problem is there are other plasma components. One of the most important things to consider is the protein corona, in a sense that when you inject these nanoparticles to the inside, they can easily pick up proteins from the surrounding to form this corona. And this corona formation is going to influence in your in vitro design, are you using PBS, or are you using the plasma from blood, or are you using serum, where the serum have huge variations that I know some people insist on using US cows versus some people use Mexico cows, etc., making sure it's the same batch. On the computational side, we are doing a very rudimental work of just simply increasing the particle size to account for some of the corona formations aspect, because size does matter, but we didn't go into it fully. So how to account for that is another challenge. So I will stop here and conclude that fundamental knowledge about biophysics and biochemistry of thrombosis are both important for directly treating thrombosis and also going beyond to treat other diseases. There's no one-size-fits-all solution for drug delivery systems because the physical chemical properties need to be optimized every single time. And then a combination of modeling and in vitro tools can help us to provide a lot of the critical information to help us move things with a maximized likelihood of clinical translational success. There are still going to be failures, but we're trying to just minimize it. Thank you.
Next set of questions before the break.
So it's me again. I was wondering when you mentioned you're using erythrocytes to carry to do the drug delivery, that my initial thought was that it's probably the worst choice because erythrocytes are an exception for basically everything when you look at different cell types. But for sure, like they're staying in the blood vessel, they have a very long lifetime. But do you think that using different cell types for the drug delivery, like the most common would be like T T cells, because they do have like this organ recognition and even cell recognition process, and they do have like this secretion process when they find the right cells. Do you think that finding another do you think that erythrocytes for thrombosis targeting is the right choice? What are the other options? And also because with erythrocytes you don't have the chance to do like gene therapy because they don't produce your drug. What's your take on that?
Very excellent point. And my short answer is I don't think comparisons across different host or carrier cells for deciding which approach is the best has been carefully examined based on my understanding of the field. And then people seem to like, okay, we specialize in the cell, like just let's do it forever. So I think that from the modeling perspective, we can predict different kind of cells. It's a good point, and we haven't really thought about any strategies here to actually maybe perhaps before we go into optimization of a specific cell type, how to design it, think about like an even more fundamental question, taking one step backward about can we actually make some first predictions about which cell type is going to be the best as a whole cell for the question that we're interested in, accounting for the fact that these different cells have different receptor ligand binding mechanisms, different cell biomechanics, size, etc. Everything is different. How to account for that? That's probably one thing that needs to be added as the preview. That's probably something that's coming back to the 30-year map that needs to be done.
Just a question for the sickle cell simulation you showed. We did some experiment based on simulation because we're like believing what the simulations were showing us, like rigid object will end up on the vessel walls. And when we did that with sickle cell, but we did that with the normal blood, the one that aggregates without washing the blood, then all this margination disappeared. My question is, did you try in simulation to add aggregation in all the simulation you showed about red blood cell flow?
So that simulation is actually not from my group. That was my Grimes group's work. We did something similar, but for malaria infection that I didn't show. So I think that it will be interesting to look at the blood setup. But one thing, like say, even the corona formation, like we didn't consider other components in blood, etc. Maybe those sort of things are also playing a role, and then we don't know. But it will be interesting to look at the data together. But I think that moving to like disease blood and then how to kind of make sure that we still have a good handle of all the behaviors as we supposed to have, that is the fundamental knowledge about moving to engineering. I think that still needs more work. So your data would be highly beneficial.
Okay. I guess I had a question about one of the challenges you talked about with these slightly leaky vasculatures in the organ on a chip formats. And, you know, when you talk to people in the vascular biology world, they are adamant that you have to culture endothelial cells under shear stresses that are, you know, mimic those in vivo. I'd just be curious, like, if you know of data, if people try to do that in these like kind of multi co-culture setups, I know they're already very complicated. And then to have to do these, you know, 48-hour, 72-hour shear conditioning makes it all the more complicated. But do you think that has something to do with the fact that you're getting maybe an endothelial cell phenotype that's a bit more pro-inflammatory, right? Because we know that low flow would be pro-inflammatory.
Yeah, so that's a great point. And then what we do is we can do similar to yesterday's speaker, if you constantly maintain the physiological shear stress, that might be too much. And also if you have just a step increase in shear rate by click on a button on your syringe pump, it's also going to be too much for the cell. So we have a very slowly increasing shear rate that we can do. And then also we found that we actually don't need to have it like a constant shear rate all the way there. So as long as you have a few hours each day to have it like a mild Q there, that seems to be fine. But that also depends on the endothelial cell type that you were talking about. Like we have two types of cells that we study, like endothelial cells. One is shear dependent, the other one is not. Like the other one, like it's not really like at least measurable changes that we're thinking about. But the application of shear rate for the culture in some systems is more for, because the one that I showed with a lot of the branch network, they have two big baths on the two sides. So they're actually, the claim of using the shear, applying fluid shear is essentially to have essentially a driving force from one side to the other. So that's not like really a shear, it's just applied everywhere. But for that system to actually really having a flow that is inside the blood vessel is also difficult. So I completely agree with you that this is a part that has been overlooked in many systems. And honestly, I think that we don't have a guarantee, even if we have the shear in the culture, whether this is going to prevent the leaky vascular issue from a problem forever, because I believe that you just need to have a single leaky point in your whole system to be destroying the whole thing. So you cannot, like, this is like a quality of your system where like you will always have maybe some cases like the seating is not super confluent, etc. So that's probably just, it's still a challenge. We'll see, we can try.
I propose you discuss that for the break, and let's thank again Maggie for this nice lecture.